इलेक्ट्रोफोरेसिस के बाद फॉर फर्दर स्टडी ऑफ डीएनए हमें डीएनए या आरएनए को किसी सॉलिड स्पोर्ट के ऊपर इमोबिलाइज करना पड़ता है और उस सॉलिड स्पोर्ट को हमने मेम्ब्रेन का नाम दिया है. Membranes are of different types which are used for southern blotting and northern blotting. These three types of membranes which are in routine use in the laboratories, they are named as nitrocellulose membrane, cellulose membrane and the most recent used nylon membrane. अब हम स्टार्ट की बात करें when southern blotting was developed, at that time nitrocellulose membrane was in use and at the beginning it was used in the powdered form to immobilize the DNA, but later on sheets of that nitrocellulose was used to immobilize the DNA. उसके बाद सेलुलोज पेपर को यूज किया गया आरएनए इमोबिलाइजेशन के लिए, but with the passage of time and use of the cellulose membrane, it was observed that the RNA is not going to give the good results while immobilized on the cellulose papers or cellulose membranes. With the passage of time, another type of membrane or paper was developed which was named as nitrocellulose paper. This nitrocellulose paper has close affinity or has high affinity for attachment of the RNA and more specifically the denatured RNA. जब हम RNA की जेल इलेक्ट्रोफोरेसिस करते हैं उसमें हम फॉर्मेल्डिहाइड यूज़ करते हैं टू डीनेचर द RNA, और इसके अलावा फॉर्मेल्डिहाइड के अलावा भी कोई और केमिकल यूज़ किया जा सकता है. In short, we can say that the formaldehyde or denatured RNA, the denatured RNA has close affinity and it binds tightly to the nitrocellulose membrane. By this finding, nitrocellulose membrane became the first choice for both the DNA study and RNA study to perform the southern or northern blotting. However, if we look at some plus and some minus points, this is still not the best choice for solid phase hybridization. अब अगर कैपेसिटी को देखा जाए नाइट्रोसेलुलोज मेम्ब्रेन की कि ये कितनी मॉलिक्यूलर वेट के डीएनए को या आरएनए को यूज स्टडी किया जा सकता है इसको यूज करते हुए, कितनी क्वांटिटी डीएनए की इसके पर सेंटीमीटर बाइंड कर सकती है. द नाइट्रोसेलुलोज मेम्ब्रेन हैज 50 to 100 माइक्रोग्राम पर सेंटीमीटर स्क्वायर capacity for attachment. Then its binding property varies as per size of the DNA or RNA. The nucleic acids which are less than 400 bases, they don't bind strongly or tightly with the membrane and they cannot be studied using this membrane. Then the attachment between the nucleic acids and the membrane is by the hydrophobic interaction, which are not as strong as equivalent bonding. So what are the drawbacks that when we proceed further for hybridization, this kind of interaction cannot withstand the high temperatures, cannot withstand the washing steps during the hybridization and post-hybridization steps. Ultimately, the RNA is going to, or the DNA is going to leach out of the membrane and the actual results or the actual signals cannot be recorded. And then the nitrocellulose membrane has another drawback that they became brittle during baking or during the processing at high temperature like 80 degrees centigrade. Therefore, these membranes become brittle and are more prone towards the breakage or towards the damage. And we cannot use these membranes more than once or twice for different probes hybridization. And once the hybridization is done, results are obtained, and the storage conditions, they are going to cause problem with these membranes. These membranes, the nitrocellulose membrane, they are going to absorb water, they are going to absorb moisture during high humidity. As a result, they can expand, they can curl, or they can get wrinkled. And if the humidity is low, they can become dry. They get charged during the storage due to static electricity. As a result, they become cracked or splitting can occur. Then there was found a replacement for nitrocellulose membrane, and this was known as nylon membrane. The nylon membrane is preferred nowadays over any other type of membrane because it binds irreversibly with the nucleic acids. It is more durable, and if it gets damaged, it is possible to repair these membranes. We can use these membranes several times with different probes for hybridization purpose. When we talk about the types of nylon membrane, with the passage of time, we modified the nylon membrane, and now there are two types of nylon membrane available. One which is neutral, having no charge. The second one which is positively charged because of presence of amine group on the membrane. This positively charged membrane is very good for binding with the negatively charged nucleic acid, which can be a DNA or which can be an RNA. And when we use these positively charged membrane, then the binding ability or the binding increases for nucleic acid with these membranes. The great affinity of the positively charged membrane for the nucleic acid gives us very good result as far as DNA or RNA is concerned for southern or northern blotting. At the same time, there is a negative point that we observe a large amount of background during hybridization period. However, this large amount of background signaling can be avoided by increasing the concentration of blocking agents in pre-hybridization during the northern or southern blotting.